I'm Alison Singer with the Autism Science Foundation, and I'm here now with Dr. David Mandell of the University of Pennsylvania School of Medicine and the chair of the Scientific Program Committee for the International Meeting for Autism Research. Thanks for being with us.
It's my pleasure.
So tell me, what are some of the most exciting studies that you're seeing at IMFAR?
Well, the most exciting studies I'm seeing at IMFAR are the ones that are breaking traditional disciplinary boundaries to really understand how genes, biology, and behavior are related and how it's going to lead to effective treatment. And we have a lot of examples of those. We have, there was a whole session this afternoon on pharmacological treatment as translational medicine. That is, how do we move past the traditional randomized trial, which tells us whether on average a medication works for a group of kids better than placebo, to understand subgroup variation. That is, the medication works for some kids and not for others. What does this tell us not only just about how we should be using this medication, but about the differences in the different types of autism? And then that leads us back to the genetics and to the biological pathways again.
There also seems to be a lot more focus this year on treatment studies in schools and on interventions in settings where children actually live and spend their time. Can you talk a little bit about those studies?
Sure, and that's my favorite kind. It's certainly my research. And we can have the best treatments in the world, and it doesn't matter if they don't make it into the communities where these kids live. And so some of the very exciting things that we're seeing in the two treatment panels that we had today and in some of the invited symposia is the increasing reliance on rigorous research designs, such as randomized trials, but then also the partnering of researchers with community organizations that are providing care to kids to make sure that whatever interventions we're developing work in those community settings.
I have heard a lot of people talking in the hallways about the keynote presentation by Dr. Jackie Crawley about mouse models of autism. Can you talk a little bit about that presentation and what it means for the future of research?
Sure. Well, anyone who comes up with a textbook that's named "What's Wrong with My Mouse" has to become every non-animal model person's favorite animal modeler. That was a wonderful presentation. She made it very clear up front that they're not trying to create an autistic mouse, which I think is most people's view of what we mean by animal research in autism. She talked very lucidly about what the correlates are between animal behavior and human behavior that indicate communication, that indicate sociability in those mice. She talked about how different genes affect those particular measurable behaviors in those mice. Two very exciting things that she talked about were the new strategies they're using to understand how mice verbally communicate to each other. The idea that mice have what we might think of, for lack of a better term, as a spoken language. And the other thing that's also equally, if not more exciting, is the idea that in many of these mice, through some kind of intervention, and we know about the biological ones, that you can rescue the phenotype, which is a way of saying that you change the behavior so that it looks like normal behavior. But something very new that she presented is that behavioral interventions seem to work with mice. So you have these mice that have a particular genetic mutation that results in repetitive behaviors and low sociability, two things we often see in people with autism, and that you can take those mice, not even when they're infants, but when they're in the mouse equivalent of childhood and adolescence, and put them with their typically developing peers. And after a period of weeks, their socialization looks just like their typical mouse peers, even though the repetitive behaviors remain. And it's a wonderful analogy for how we should be thinking about treatment for human children. We often think about these biological interventions and these psychosocial or behavioral interventions as two completely separate domains. But what these mouse studies are showing us is that what medications might do is increase people's readiness to learn, but they don't provide learning. And where the learning is going to occur is through those behavioral interventions, and not just one-on-one at a table in a room with a therapist, but through the socialization that occurs through one's peers.
It also seemed this year that there were a lot of studies that fit into that pathway that you were describing, where we start with the genetics and then we move to the animal models that Dr. Crawley described, and then we move to more treatments. Do you see that? Do you see more studies that are along that continuum?
This is a really important shift, I think, that we're seeing over the course of the last couple of years, and that is really manifesting in the studies that are being presented at IMFAR. We used to have a lot of genetic studies that seemed like genetic studies for the sake of genetics. That is, you would get the biggest group of people with autism together that you could find. You'd take their blood or their spit, you'd assay their DNA, and you'd look for the different genetic hotspots that might pop up. And every time we did these studies, it was a different genetic hotspot that would pop up. And I think that it wasn't really clear to families because scientists weren't doing a very good job of talking about it, why we care. What is it going to mean if we find this new gene that's associated with autism, but it's only associated with a certain percentage or small percentage of kids with autism? What we're seeing this year is a much better strategy where once a gene is found, scientists are then taking that gene and really trying to understand what it does. What are the correlates in biological pathways? What are the correlates in the brain that are associated with having this particular genetic difference? One of the great places we saw that at this meeting was in the Neuroimaging Genetics Symposium, where people are using genes to correlate with what's happening in the brain using functional magnetic resonance imaging, or fMRI. And so we see that among people with autism, those who have one kind of genetic difference, their brains light up in one way, and kids who have another genetic difference, their brains light up in a different way. But in both cases, when we look at the behavior looks at autism. Well, now that we're understanding the correlates between the genes and the brain, the next step is to understand the correlates between the brain and behavior. And when we do that, we're going to find that there are really different processes in the brain that then occur, and then create something that looks very similar. And that's going to help us understand how we should be focusing our treatment and how we should be doing it differently for different kids.
So I'm going to ask you to look into your crystal ball now and say, what do you think we'll be talking about at IMFAR 2011? What will the key themes be there? What types of studies will we be seeing then?
So I think we're going to see two kinds of studies that are really a continuation of many of the things we've seen here. So one of the big studies and stories here, obviously, is the idea that for children whose autism seems to result from a specific genetic mutation, that is Fragile X, we may have a medication that comes as close to curing autism as I think we've been. The next step is to see if that same biological pathway is occurring in other kids with autism who don't have Fragile X, but it may be the same process that's been affected, and whether the medication works for those kids. So I think that's going to be one big exciting story next year. The other thing that I hope we begin to see is we are having the development of all these efficacious interventions. Efficacious means that it works in the lab under ideal circumstances when delivered by highly competent therapists. The next question is, how do we take those and move them out into the community? So for instance, the study that we're doing with the School District of Philadelphia, where we're trying to take proven efficacious interventions and see whether in a large school district we can make the kind of organizational and structural changes we need to make in order for that intervention to be delivered effectively. I'm hoping that we see a lot more studies like that, and that it leads us to think about changing local, state, and federal policy around how we deliver care to people with autism, how we train the professionals who are working with them. So next year I'll be very excited if those are the two pathways that we see.
Well, I just want to thank you for an amazing conference, a tremendous amount of learning, a tremendous amount of collaboration, and thanks for being with us today.
It was my pleasure. It's been an education for me too.